Look to the Future session, which is on genetic interventions for neurodevelopmental disorders. And we are beginning with a talk about the science from Dr. Wendy Chung, who is the Kennedy Family Professor of Pediatrics in Medicine at Columbia and Director of the Clinical Genetics Program here. And she is a clinical and molecular geneticist with extensive experience in ELSI research as well as her basic science research. Thank you, Wendy.
Thanks so much, Ruth. I'm really pleased to be here today. Hopefully everyone can see this in full screen mode. Is that working, Ruth?
Yes.
Okay, very good. So as I'm doing this, as Ruth said, I'm a practicing medical geneticist, and I'm going to bookend some technical things in the middle with two real life cases, or at least two real life conditions, to give you a sense of literally as of today where things are. And then in the middle, I'm going to go through some technical aspects of some of the genetically based therapies, but hopefully in an accessible way. I'll try not to make it too technical. So the first is really a success story, and it's for us in the field what's gotten us very excited about the possibilities of what may be possible for many conditions. This is a neurological condition called spinal muscular atrophy. As the name implies, it is a loss, progressive loss of motor neurons, and it used to be, I'll underscore past tense, used to be the most common genetic cause of death for children less than 2 years of age. It's a condition where the children are born normal or seemingly normal, at least with type 1, 2, or 3, and then undergo a progressive decline where the major symptom is muscle weakness. So they get profoundly weak with the type 1 and the most severely affected infants, to the point that their diaphragm, their breathing muscles, is so profoundly weak that they're incapable of sustaining life. That is, that they literally cannot breathe and catch their breath. There are those individuals, type 1 babies, usually die by 1 year or 2 of age, and children and adults with type 2 and type 3 are less impaired, but still eventually lose the ability to walk independently, and many of them are wheelchair bound. Importantly, cognitively completely intact. So very lovely people and very bright, many of them quite accomplished in terms of from their professional point of view, but with a disability largely in terms of mobility. So this particular condition from a genetic point of view is unfortunately not something that we can do for every gene, but this particular gene in gene evolution has undergone a gene duplication event. That is that the underlying gene survival motor neuron 1 in humans has been duplicated to have another copy of itself, survival motor neuron 2. This is relatively recent, so mice, for instance, don't have this. The SMN2 gene is good, but it's not perfect. And so in fact, this is a little bit too complicated. You can just listen to what I'm saying. But for individuals who are born with mutations, and this actually 2 mutations for this recessive condition for SMN1, they still have oftentimes a backup copy of SMN2. SMN2, though, only produces normally about 10% of the protein due to some very relatively mild changes, but ones that affect splicing, such that there's deletion or non-inclusion of exon 7, and subsequently from each one copy of the SMN2 gene only produced about 10% of the protein. Individuals with SMA can have more than 2 copies of SMN2, in fact, could have 2 or 3 or 4. And in fact, there's kind of a gene dosage effect. Knowing about that in the natural history allowed Adrian Kramer at Cold Spring Harbor to think about, could this be manipulated? That is, could you use SMN2 and I won't go through the details, but using an oligonucleotide to trick the gene into being spliced so that you would get inclusion of the exon 7 and in fact full-length protein, and that full-length protein, again exactly the same survival motor neuron protein, and be able to compensate. And in fact, this was taken on eventually beyond Cold Spring Harbor and was commercialized. But an important part of what I want to say is that this required really both diagnosis and therapeutics. And I'm going to emphasize this, that the diagnosis gets you to the starting line, and then the next phase that we're talking about is really getting to the therapeutics. And one of the challenges for SMA was that this was, as I said, a degenerative course. And many individuals weren't diagnosed until potentially beyond the therapeutic window, or at least the optimal therapeutic window. So Kathy Swoboda had thought about this earlier, and then I took this on in New York in 2016. The idea that you might be able to do newborn screening, which is currently the largest public health genetic infrastructure that we have, and take one of these blood spots that we take from the newborn heel stick that we do for things like phenylketonuria, but take that and do a genetic screening test for SMA. This particular condition, I want to admit, and we thought about this quite carefully, was quite amenable to this technology because there's one single mutation that in fact 95% of individuals with SMA had. So it's something where you can scale. And in fact, I'm very proud to say when we did this in New York, we got the cost per baby down to 21 cents per baby. So we got the diagnostic side of this down very, very inexpensively. And it is a condition that is essentially 100% penetrant. So some of the questions that come up as we think about, well, are you talking about a risk factor? What are you talking about? In fact, we can, with relatively good fidelity, predict both that someone's going to have SMA and even to a large degree how severe the SMA is going to be. When we started this pilot study in 2016, it was interesting because Spinraza, or this oligonucleotide, was at the time in clinical trials, and it was not FDA approved. We didn't know it was going to work. We didn't know how effective or durable it would be. We didn't know if there'd be toxicity, but for this condition that otherwise had no treatment, it was felt that this was something that we should, again, try and think about newborn screening, in part because identifying these newborns pre-symptomatically would help get them into the, if they chose to do so, the clinical trial, but at specifically a time frame when they might have the greatest chance of benefit. And so in fact, we started this, as I said, the first, I'll show you a video for her, the first, we had children that were identified through this with SMA, and they weren't coerced, they weren't automatically assumed they would be in the clinical trials, but they were offered enrollment, and in fact did decide to do that. And this is one of my favorite videos. This was the first of our children who entered, was identified from newborn screening, entered that Spinraza clinical trial. And these are her home videos. She would have been predicted to have type 1 SMA and not live to, at least not to her second birthday and probably not to her first birthday. But as you can see, at 14 months of age, she's strong, she's walking, she's meeting her milestones at the regular prescribed time. And I'm very proud to say she continues to be a healthy, now actually first grader. So she's doing very well. And this really has propelled both the FDA approval for the oligonucleotide Spinraza. It propelled us to have the data to make the case to put SMA on the recommended universal screening panel, so that this is now nationwide adopted as part of newborn screening. And as of yesterday, 95% of babies, in fact, are being screened, and there are 3 lingering states that are coming on board. And in fact, it is not just Spinraza that is now FDA approved, but in fact, there are 3 FDA approved treatments for SMA, which includes a gene therapy, which includes being able to do, theoretically, a one and done intravenous infusion of a gene addition to be able to compensate for the missing survival motor neuron protein. So with all of this, that's the real excitement in the field. This is, transformational in terms of the impact. For me, from an equity point of view, it's incredibly important because this allows every single baby an equal chance to a good start to a healthy life. And this is best case scenario, though. So I want to bookends that at the one end to realize this is theoretically what's possible. I also want to say that as I'm talking about the therapies that I'm talking about, I'm very cognizant that even despite all of the work that I and people like me have done in terms of gene discovery and trying to understand these conditions, when we speak about neuropsychiatric conditions, I'm not saying that every condition needs to be treated, every condition needs to be cured. I do think, and I come from, as Paul mentioned, the area of autism. As we think about the complexities, there are complex nuances. And so I want to be very clear that the things I'm talking about in terms of treatments are for individuals, number one, who choose to have treatments and who feel that for themselves and improving their lives, that's helpful or necessary. And that really what I'm talking about are largely highly penetrant monogenic conditions associated with really, really severe impact in terms of quality of life. I'm not talking about someone who has a little bit of depression, or who may be a little bit, sort of have some social challenges on a milder end. I'm talking about things associated with really significant impact in terms of daily life. So this is just showing you the landscape in terms of gene therapy. This is meant to show you with individuals who are developing, pharmaceutical companies, biotech companies who are developing new treatments. These are the small center here in gray, are those that are actually have made it over the finish line in terms of something that's marketed. It's still a limited number, as you can see. And in particular, I've highlighted in terms of those with stars, things that are related to neurodevelopmental conditions. And so you can see across the landscape, even the things that are now FDA approved might not be what you would think of, but for instance, for eye diseases, where we can have a very sort of small part of the body where we need to focus this. There are things that we certainly do in the area of cancer that are related to this. But again, when you see what's come over the finish line for the types of things our CIR has been focused on, we're not over the finish line yet. So this is the future state that we're talking about in those possibilities. So within this context, I wanted to give you a bit of primer just so that it's scientifically grounded as you start to hear some of the pros and cons and what's possible and not possible as we're thinking about this. And it is, like I said, with the assumption that you know the target. I will be clear that, and I'll explain this a little bit more, many people think about a specific genetic susceptibility that someone has, a particular genetic factor, and being able and having a broken gene, so to speak, and having a way to fix it or having a way to compensate for it. And I think that's what we're talking about mostly within the context of this morning. There is theoretically another use case where you're not targeting specifically that particular gene, but you may be thinking about the pathway that's involved. We think about this on the cancer side in particular, but there's a pathway involved, and you're using any of this gene therapy strategy as a way of delivering a drug. You can think about it that way. The drug may be essentially a protein that you're doing, but it doesn't have to be the sort of genetic difference that, or the genetic variant that the person was born with, that you're targeting as you're doing this. I'll also be clear that as I'm talking about this, we're not thinking about germline gene therapy. And for this ethics group, I know you know the difference of what I'm talking about, but we're really thinking about doing this somatically. This is not something that's transmitted to the next generation. We're not thinking about altering the germline. We're not thinking about enhancements as we're doing this. This is really thinking about, again, significant negative impact in terms of quality of life and how to be able to make life better for those individuals. So with this, there are many different ways to package this material as we're doing it. I'll get into a little bit of the detail. Many of you, for instance, have even experienced this personally in terms of when you had your vaccine for SARS-CoV-2, the same packaging that was used in terms of delivering that RNA is to a certain extent some of the same packaging that at least is being trialed right now. I can say we're running one clinical trial using that same technology right now for glycogen storage diseases, a metabolic disorder. But there's everything from that type of delivery system to others that are trying to use viruses to be able to deliver, again, a durable way of being able to get in that gene or those nucleic acids. Others that are based on oligonucleotides, as I was saying for Spinraza, where it's a way of being able to have short amounts of DNA that go in and that may affect splicing, for instance, or may knock down a gene. And so there are other technologies and other companies that are thinking about that. In some cases, I call it the poor man's version of gene therapy, but we do think about bone marrow transplant, where we may not be altering the gene per se, but a poor man in the sense of using someone else's genes. And certainly that's been very widely used for things like leukodystrophies or lysosomal storage diseases, in addition to some other things we use, but that may or may not affect the brain so well when we use things like enzyme replacement therapy or simply infusing the enzyme that's missing. So within this, there are a lot of key questions that we ask ourselves when we're thinking about these therapeutic strategies. And one of the most challenging for what we're talking about today is where in the body we need to get it. we clearly need to think about the brain or the central nervous system, and that's quite challenging, I will say, across the gene therapy aspects. Being able to get in the brain, being able to get equally across the different parts of the brain to the deep nuclei. In some cases, what happens when you don't do that? There's another critical question for many, and I will say in some cases, although not all cases, we need to get beyond the brain. There may be certain circumstances in which these conditions actually have peripheral effects on the heart or another part of the body, and so we need to know that. A very key critical question is, when in life is this process evolving? So as I was talking about with SMA, is this something that's a degenerative condition? Do you need to get in before the degeneration occurs? Do you need to get in before people are having symptoms? Is this a developmental condition, on the other hand? And in those cases, when is development occurring? And do you have a window of opportunity in terms of treatment that may only extend in some cases in utero? Do you have some cases where it may extend for the first few years of life, but where efficacy may not extend throughout the entire life course? So critical to understand that natural history, and in particular reversibility. So in certain cases, we wonder if there's damage that's been done, if there's, like the case of SMA, if there are neurons that are lost. They're probably lost forever. And how reversible that is there any regenerative potential? In many cases, the answer may be no. And so again, when we think about that, understanding that if there are other cases where there's accumulation of toxic buildup in neurons where they may still be alive, but they may be damaged, can any of that be reversible? And in some cases, we have hypotheses based on animal models, but we may not always know. One of the challenges to do this at scale, realizing that there are 7,000 rare genetic conditions and over 1,000 rare genetic conditions affecting the brain, is to understand the mechanism. And you'll understand a little bit better what I mean by that in a second, but we have to understand whereby the exact genetic variant actually causes disease. And in particular, there are a few mechanisms that we focus on, but is it that the gene is not working in the sense that it's lost its function and we need to replace it? Is it the fact that the gene is doing something it shouldn't be? It's got what we call a gain of function or a dominant negative effect where we need to get rid of it because it's doing something it shouldn't be doing. Or is it potentially just changing its function, perhaps changing the substrate for receptors, for kinases, for phosphatases? Is it changing the substrate and then affecting a cascade of things downstream? And that becomes very important to think about which of the strategies we will eventually use. So as we do this, a lot of the work, just so that you know, the science that's done oftentimes is done first in vitro in cell-based systems, oftentimes using animal models to progress from that, and of course, eventually in people. And for a lot of what's going on now within the field, a lot of it is still at the cell-based level. And as we're doing this, many of the cells that are used are in fact derived from patients. Improved modeling and being able to use induced pluripotential stem cells and differentiate them into a variety of different cell types within the brain has helped the field, but is still obviously not a replacement for doing in vivo studies and clinical trials. I'm going to go through these different strategies, but just to say that as I was talking about, there are different strategies for doing this. I'm not going to worry so much about the delivery method, whether it's a liposome, whether it's a different vector, what types of vectors to use. There are all sorts of details we could go into, but I'm going to keep this at a very high level discussion. So as I said, one way we can do this is gene replacement. So this is where it comes in. Is the problem a loss of function? Is the problem that the gene, in fact, is not working? And there is a very fine other point to this, which is in some cases, the genes can be dosage sensitive. So the issue is that it's sort of the Goldilocks problem. Too little, if it's too cold, it's not good. If it's too hot, it's not good. You need to be just right. And so there are many conditions where there is problems with a loss of function and you'd like to do the gene replacement, but you have to be very careful to not overdo it. You need to get it just right in terms of being able to do that. And increasingly, there are technologies that are evolving. I think of them as thermostats to be able to get it just right, but but it is a critical issue because you can trade one condition for another condition if you don't get it just right in some of those conditions. There are also fine points about what I was alluding to. It may be that you could replace a gene or add a gene because maybe it's something that by doing it, you could compensate. It may not be the gene, the genetic variants that you're directly attacking that the person was born with, but this may be a compensatory mechanism, and this therapeutic strategy is simply to be able to add. It's sort of a delivery system. It's a way of getting the medicine in, but in this case, the medicine is a gene or its product that's produced. And so again, this sort of opens us up, and people think about this a lot, especially, as I said, with cancer and some cardiovascular diseases. When the mechanism is that the gene is doing something you wish it weren't doing, you actually need to silence that gene. You need to get rid of it. In some cases, you need to get rid of the gene entirely. So if the gene has 2 copies, as with autosomal genes, you may be able to knock down the gene entirely and just get rid of it completely. In some cases, based on the time over the life course when this gene is necessary, it may be that it's not necessary anymore at that life point, and so you can just get rid of it. There are other times when you have to be quite careful, and this is true especially of heterozygous or conditions where there are 2 copies of the gene, and it may only be one of the 2 copies of the gene that's taken on this rogue function. You may have to be careful in those cases because in fact, completely knocking down or getting rid of both copies of that gene could be detrimental. It's again kind of like this Goldilocks problem, and I won't get into all the vagaries, but what I hope you're getting the sense of is it's complicated. It's not that there's one size fits all, and it's not that it's trivial. You really have to understand the biology. You have to understand a lot of foundational, sort of very basic work about these to be able to think about the right therapeutic strategy. And the reason I say this is because it takes time. And I think I'll get to this at the very end, but there's oftentimes this sort of anticipation that, the gene will simply fix it. And I want to sort of set the stage that it oftentimes takes decade or more in terms of getting that foundational knowledge. So one of the most exciting ways that some people have thought about is gene editing as a way of more universally being able to go in and correct the problem. That is that if you had kind of an eraser that you could use, and if you knew exactly what the gene was, you knew exactly what the nucleic acid was, could you go in and just kind of erase what the problem was and pencil in what the right sequence is? Could you do this in a universal way in terms of gene editing? And in fact, there are many technologies that have been developed to be able to do this. I again won't go into all the details. There are still issues in terms of the editing efficacy. Do you correct all of the cells? As I alluded to in the brain in particular, you may need to get to every single last one of the cells for a brain condition, whereas for a liver condition, maybe you don't need to get to all of the cells, maybe 20% of the cells is just good enough. So we have a higher bar, a higher standard when it comes to brain conditions. In particular, we also worry about promiscuous effects. That is that with some of these technologies of gene correction, it may not just hit the target of what you're trying to erase and being able to pencil in. It may do something off target. It may do something that, for instance, could activate a proto-oncogene for a cancer. It may do something that would break inadvertently, but break another gene that would have consequences. And so doing this with very high fidelity is necessary before we start getting into this in large way in terms of doing it in people. So as I said, many people know, for instance, that the Nobel Prize was awarded for CRISPR-Cas9 system. This whole system was not really developed specifically for gene therapy, but it is being used increasingly to think about that. It's a way of being able to go in, as I said, and in a very focused way, target where the specific variant of interest is and be able to go through and make a correction. There is a whole delivery system that needs to happen to be able to get all of the component pieces to the CRISPR-Cas9 system, and then the guide RNAs in terms of the right place and location to do this. And people are thinking about this initially where it can be done ex vivo. That is, for instance, when it comes to things like taking stem cells from the bone marrow out of the body, being able to do some corrections, making sure that you target the right variants and don't have those promiscuous effects, and then can put that back into the body. And so as gene therapy is going forward, it's certainly going forward in situations where there are a smaller target number of cells that need to be done, and where there are ways of monitoring this really outside the body rather than doing it inside the body when you get potentially one chance to do this and can't take it back, no backsies. In doing this, there are some concerns about that technology because it does make something called double-stranded DNA breaks. And I won't go into it in a lot of detail, but as that happens, can inadvertently create some other problems, some other types of mutations. And so there are other technologies evolving where there are single-stranded DNA breaks that have greater fidelity, less off-target effects, and also technologies being evolved that can target a large number of different types of mutations. So things that might be not only single nucleotide differences, but quite a large repertoire, and that offers some potential that goes beyond some of the early systems that were started. As I said, there can also be nucleic acids themselves. This Spinraza, as I was talking about, or allele-specific oligonucleotides. That's one thing that right now in the current sort of thinking about this has some attractiveness in the sense that when you start doing this viral delivery system, I alluded to it, but you get one chance in terms of doing that. You oftentimes, based on the immunological response to that, can't do repeated dosing. And so if you get it wrong in terms of either not doing the right dose, perhaps not doing too low a dose of this and not getting enough of the gene in, you don't get a second chance to go back and correct that. And so people have been thinking, and then there are the off-target effects, like I said, that some people are quite worried about. And then there's a whole issue about the body's immunological response to that. And I'll be very transparent. There have been some deaths in our recent past in terms of gene therapy for some muscle diseases. And so there's some concern in terms of safety issues as well as efficacy issues, despite all the excitement, again, that I talked about with SMA. So some of these other delivery systems, ASOs, for instance, or liposomes, people think about this as not being one and done. That's the advantage, and that's the disadvantage. There is the ability to be able to adjust the dosing if there's toxicity, to pull back on the dosing if there's not sufficient efficacy, going up on the dosing. That's the good thing. They're also relatively scalable in terms of manufacturing. There are facilities available to do that manufacturing. And believe it or not, for gene therapy, that's one of our rate limiting issues right now, is just not having the factories to the grade that we need to make clinical grade vectors and viruses to do this. So that has some advantages. The disadvantage is that it's not forever. And so when we think about delivering this, in particular, delivering it to the brain, do we have to do like what we do with chemotherapy in terms of cancers in the brain? Do we have to repeatedly deliver chemotherapy, or in this case, an ASO, to the brain? Do we do it through the back, as I'll show you in a second? And this is invasive. And for certain people, they may need sedation for these procedures. They may be associated with side effects. So this gets complicated quickly. There are, as I said, other ways using these liposomes. Certainly, we've had a lot of experience now with the Moderna in particular has had experience with this, and I think is refining what they've been able to do based on some of the vaccine experience. So with this, the idea with the liposomes, as I was saying, is these are packaged in lipid particles. Those lipid particles fuse with the cell membrane and then can deliver their cargo, in this case, nucleic acids that can then get into the cell. Other delivery systems are viral systems. Adeno-associated virus in particular has been a good way to target specific cell types so that we can get greater specificity. This allows us to minimize the dosage that we have to use. And again, higher dosage can be associated with an immune response to the virus. So we like being able to use a lower dose where possible. And there are specific AAV subtypes that we can use for tropism to the brain. And as I said, being able to do this, so you might not have to do direct injections to the brain, in some cases be able to do even peripheral infusions. As we think about doing this, the virus itself has been essentially inactivated so that it can't continue to replicate. It can't sort of go rampant into the public and suddenly infect everyone like SARS-CoV-2 is doing. So we've gutted the virus from having its usual replication machinery and replaced it with what we need to in terms of the gene addition or gene replacement. There are limitations, though, in terms of the cargo, the size gene that can be packaged into these viruses. viruses. And so there are many genes that actually cannot be packaged. And in particular, many brain genes are quite large. That's one of the characteristics we see of many brain genes, and many of them are too large to be packaged. People are working on splitting these and having them sort of in the cell come back together. And so there are technologies that people are trying to use to address this. But like I said, it's not completely worked out for this. So when we think about this in terms of the clinical trials going forward, we do these first in animal models. So looking at toxicity when we can, looking at efficacy, non-human primates oftentimes used in terms of a step in terms of doing that, but you really cannot predict some of the things that will happen until you do them in people. Safety is paramount for us. And we think back to Jesse Gelsinger, who was treated 3 decades ago, actually. I think I've got that right. 2 decades ago, at least. We're trying to avoid for the field what was an unfortunate misstep early on. But there's still, this is not completely worked out in terms of all the things we want to do. And so we're going slowly into this space to make sure we don't trip or cause problems that are either lethal or problems with long-term side effects. I will say that in the eye diseases in particular, that because you can, again, do the treatments in such a small region, there has been success in terms of doing that. And I do think that's a continued area. Deafness, believe it or not, is now also an area, again, confined hair cells where we need to deliver the treatment, but to do it in a very focused way. So as I said, there are real challenges when it comes to the brain specifically. And so, we think about the risk and reward, and we're trying to really focus on things that really do have significant burden because this is not easy to do. There are different ways to deliver to the brain. I'll show you in a second. We do things through a lumbar puncture into the spine, knowing that the fluid, the cerebral spinal fluid, bathes both the spine and the brain. We do all sorts of maneuvers to try and get the flow going more up to the brain when we need to for that. But there is concern about patchiness of delivery and whether or not, when we think about networks of cells in the brain, whether or not if you disrupt those networks, maybe you treat some cells but not all cells, could that even be worse potentially than if you treated no cells? I don't say we have the answer yet, but we are trying to think about that quite carefully. And as I said, when we think about reversibility, we are trying to think about risk and reward, and it comes up to the SMA issue again, which is that early diagnosis may afford us the opportunity for our best chance at treatment. And so I think of the infrastructure that's necessary to support early diagnosis, realizing that for many of these conditions, they're due to de novo genetic variants, that is, ones that are new to the person, so you can't rely on family history to be able to identify those families or individuals at risk. As I said, the immune system can be powerful, and that is oftentimes where we see the deaths or due to liver problems. There's the possibility of targeting the wrong cells, and as I said, there's the possibility of disrupting a protective gene that protects against cancer that can be a problem. So we're quite cognizant of that. With this, I alluded to this already, but this is something where we oftentimes think that we've got one chance, and everyone may not have that chance. Individuals, if we use a viral vector and a virus that people have already been infected with before, they may have antibodies that preclude us from using that same virus for them again. And again, it speaks to over the life course, earlier is better. Earlier, we have fewer individuals with antibody to those viruses that we might use, and so we've got a better chance at treatment the earlier in life that we go. Just to say that this is going on in terms of many different studies right now. One of the most active areas is Angelman syndrome. There are actually competing clinical trials right now ongoing for Angelman syndrome. We're waiting to see whether or not some of the earlier studies from 2020 that showed some promise yet some toxicity, how those will go forward. There have been some disappointments also in the field for Huntington's disease, unfortunately, recently, but there are many other, both, as I said, brain as well as non-brain clinical trials going on right now. I'll end with thinking about an area I've been working on recently, which is really now at the extreme for an N of 1 situation. So many of these mutations are, in fact, if they're not unique in terms of the entire global population, they're unique at least in terms of what we know about. And I work on a condition called KIF1A-associated neurological disorder. It's a degenerative condition, which is just really cruel in terms of being being lethal for many of the individuals in our community. And we really need to be able to get in earlier. And it suffers from just the inconvenience of having many, many different mutations. I'm showing you here what are many, oftentimes unique mutations in the motor domain, but even in the more common mutations, no one mutation accounts for more than 5% of our community. And so if you have to do something that we're specific, you have to think about many different potential ways of treating this. This particular mechanism is due to a dominant negative. These individuals are heterozygous, and we have 2 copies of the gene, but we need to get rid of 1 copy with the mutation, oftentimes a missense mutation, which is kind of gumming up the works. And so the strategy that we've been thinking about is an ASO strategy, where you can essentially be able to knock down in an allele-specific way, hopefully, this 1 particular copy of the gene using RNase H within the cell, but yet still spare the normal copy of the gene. And it is critical, I'll say, that we spare the normal copy of the gene. We don't have the benefit of time in this condition, that for some of the children, their life expectancy is a few years. And so in particular, there's one of my individuals, one of the patients I treat and have been treating for several years, where we are going to be going forward with an N of 1 treatment. Tim Yu at Boston Children's has done this with Millicent and some other cases as well. Neil Schneider here at Columbia has done this with FUS ALS. So there's some precedent. We're not the first by any means to try this. But we are going to be doing this for this 1 condition, but also for other conditions that are in the works in collaboration with NLORM. With this, just to give you a sense, we have to repeatedly dose these individuals by giving this infusion into the spinal cord here. And these individuals are of a neurological status and an age where they're not able to cooperate with this. So we do have to give them sedation. And we expect that the treatment will be repeated dosing approximately every 3 months or so. And with this, there's always some risk that comes with every treatment. And so, as I said, this is something that we don't take lightly as we think about this. And on balance, we think about whether or not, if conditions in fact are lethal, and if there are no other alternatives, that being a case where there might be relatively more benefit. So as we're thinking about this, there's a lot that I think we're going to have quantum leaps forward, is the way I'll describe, for brain conditions. I don't know what is going to be next after SMA, to be frank. I think a lot about this, but I do think there are opportunities to scale this using lessons learned. And so as we're starting to gain some of those lessons from some of the more severe conditions, also thinking about the infrastructure that's necessary for early diagnosis, understanding these conditions, understanding equitability, how to make sure everyone gets to that diagnosis. And if we do have treatments, making sure that's also equitable, that these aren't million-dollar treatments that only a few people can access. So I will stop there.
Thank you so much, Wendy. It's really a new world. We have time for some questions. Again, you can ask a question in the chat, or you can raise your hand. Dr. Lewis.
Hi, thanks so much, Wendy, for that talk. I'm wondering what you think the implications are over, say, the next 10 years for the genetic screening that we need to do. Like, where do we need to get to on that kind of timescale?
So I'll say something somewhat radical, to go back to the spinal muscular atrophy. So for some conditions, I'm not saying all, but for some conditions, we know family history, and that's enough to be able to give us that information, and people can make their own choices about when they want to get diagnosed or not. And I think that's one bucket of conditions. There's another bucket of conditions, and I'm a pediatrician, full disclosure. And as I think about children, some of those conditions, as I said, due to de novo mutations, you don't have the benefit of family history. And there especially, I do think we have lots of limited windows of opportunity. And I think about scalability and equity. And so I think about how to use what we did with newborn screening for SMA, and how do you potentially, for certain conditions, I'm not saying everything, but for certain conditions, might we be able to use a sequencing-based method for newborn screening. It's interesting, though, because our paradigm for newborn screening is that we don't add anything to newborn screening unless we have a treatment, and we know the treatment is efficacious. Those are our WHO criteria. But on the other hand, it's a bit of a, as I showed for SMA, it's, these two feed into each other. If you can't get early early diagnosis, maybe you can't get to a treatment. And if you can't get a treatment, maybe you can't do newborn screening. So I think we're going to go through some pilots in which we give people the ability to fully consent and fully think about if they'd want to know this information for either a newborn or a child or an individual across a lifespan. I sort of, say that there are those opportunities, and decide for themselves whether they want to know this information, knowing that we can't do it for everyone or every condition, but we'll need to gain some experience doing this and doing it specifically across individuals of diverse ancestry so that we can do it equitably.
Other questions, comments?
If I might ask something else. Oh, sorry.
Okay, go ahead. Yes.
Well,
okay.
What about so you just made an argument for very targeted for specific genes. What about some of the antisense technologies which are might potentially be useful for particular variant classes and many different genes in the de novo status, which might necessitate something broader than a gene targeted approach? Yeah. What are your views on going whole genome?
Yeah. So anyway, I won't go to your technical point, which I know you know about, Anna, but there are interesting things about certain technologies that are agnostic of the gene and simply by mutation type. And so I didn't get into that detail, but that's true. So I think I'll just sort of put it out there. So one can think about the infinite flexibility I think you're alluding to. Rather than doing a focused analysis of, say, 20 genes, you could say, well, let me do an analysis of the entire genome, put everything on the table, have that as infinite flexibility, and then you can interpret. You can look at what you have ability to look at based on what technology from a therapeutic side, what you can do something about. And so you could think about whatever modality, whatever research study you want to run. But I want to emphasize this is a research study with people consenting in to do this. You could imagine doing this where you do exome, genome, but something that gives you flexibility. And you can also think about how you would engage the person to potentially update, I'll be radical, update consents over time, be able to, as therapeutics change, as things change, dynamically be able to grow as the situation changes. And if we could think of a future state where we could support that, I think that would be amazing.
Great, thanks.
Dr. Pierce.
Thank you. Thank you for the presentation. It's a fascinating presentation. My question pertained to a chart that you showed in which you listed one of the disadvantages for a couple of the interventions was that it's not permanent. And so one of the things that you've stressed is that the developmental phase may be particularly important. Is there a possibility, or maybe you talked about this, that it not being permanent could actually be a benefit? And are there instances where, do we have an understanding of what the risks might be for a non-permanent intervention in some circumstances? Thank you very much.
No, that's a great point. So I'm embarrassed to say this, but there's just so much that we don't know. So we make extrapolations based on gene expression data to think about where in the life course and what cell types and what part of the body we think might be critical. And there are certain conditions where we see expression of the genes heavily during fetal development or early childhood development, and then it's turned off later. And I think that's what you're talking about. Exactly. Maybe you want to, treat, but you only want to treat until age 2 or something like that. And then you don't have to run the risks and you can just cut it off and you've got what you've got at that point. And that would actually be wonderful because you don't have to worry about all of those promiscuous off-target effects that I mentioned. I think the number of conditions like that, the tricky part is the genes that behave that way tend to also behave where they're very, very active during in utero development. And I haven't even gotten there in terms of talking about in utero gene therapy. There are people that are already thinking about that and trying to consider that, but that then pushes the envelope back for us about when in time. Now thinking about during a pregnancy, like when would you have to get that diagnosis and can you do it invasively or non-invasively? And so it changes the conversation quite a bit, but there are people starting to think about just that and what's necessary to be able to get to that stage of life should it be needed. But it's very complicated from an ethical point of view, as this group totally appreciates.
Perfect. Thank you so much.
And thank you very much, Wendy.